So, hello students, let us have a quick take on classification of blood, which is a part of the human circulatory system. So, you have a chapter called as human circulatory system in biology, and let's have a quick discussion on classification of blood. So, very interestingly, blood can be classified into the formed elements and the plasma. So plasma is, you can say, the liquid form of the blood, and then we are having the formed elements, right? Formed elements are your blood cells. So basically, if you have a test tube in which if you keep blood, so blood on standing, okay, blood on standing will convert into two layers. One, the upper layer will be plasma, and the lower layer is going to be the formed elements, right? So plasma is what? It's a straw colored fluid. I mean, plasma is also consisting of serum, which is a straw colored fluid. So plasma is there, it's the blood without the blood cells. So what is plasma? Plasma is the blood without the blood cells. So if you remove the blood cells from the blood, after the blood cells clot, what is remaining on the top is plasma. So it's the liquid part of the blood, okay, and which is consisting mainly of water. Then plasma proteins, if you remove the plasma proteins from the plasma, it will convert into serum. So if you want to write what is plasma, I can write like this: plasma is equal to blood minus blood cells or the formed elements. Then what is serum? Serum is your plasma minus plasma proteins, a straw colored fluid. So plasma minus plasma proteins is nothing but serum. So what's there in the plasma? Quickly, water, plasma proteins, then we are having glucose, all the dissolved part, all the dissolved nutrients of the blood. So amino acids, okay, and all other nutrients which are soluble in the blood will be present in plasma. Okay, so water, plasma proteins, glucose, amino acids, then the hormones, vitamins, all that can dissolve in the blood will stay in the plasma. All that can dissolve in the blood can stay in the plasma. Okay, will stay in the plasma. So now what are formed elements? Now let us focus on formed elements. So formed elements can be further divided. They are the blood cells. They can be further divided as the RBCs, which are very well known as the erythrocytes as well. You can also call them as erythrocytes. Okay, we have to discuss in detail about the RBCs or the red blood cells or the erythrocytes. Then we are having leukocytes, which in other terms is called as the WBCs, white blood cells. Okay, so you must have heard about the term leukemia. Leukemia is blood cancer, wherein there is, you can say, excessive number of cells, excessive number of white blood cells. So when there is excessive number of white blood cells, what's going to happen? It's going to cause leukemia, which is called as blood cancer. See, always remember mia meaning blood, and leuke is the leukocytes in blood. So excessive leukocytes in blood is called as leukemia. Okay, then deficiency of RBCs in the blood is called as erythrocytopenia. What it is called as? Erythrocytopenia. Erythrocytopenia. It is the deficiency or decrease in number of RBCs. That is erythrocytopenia. Well, and the third blood cells are thrombocytes, which are also called as the platelets. Thrombocytes or the platelets. Okay, so here we are having the formed elements, RBCs or erythrocytes, which are now having a lifespan, okay, lifespan of 120 days. Who? The RBCs are having a lifespan of 120 days. So if you are given the choice of death, like you decide on which day you have to die, or if you are told that at the age of 80 you yourself you go and die, right? But some people wouldn't prefer dying at the age of 80. Even at the age of 80 they'll feel like living for a little more longer. But it's not the case in about the RBCs or about the cells. Now cells are having something called as autolysis. Okay, so after 120 days, the cell is going to undergo autolysis, which is also known as programmed cell death. Okay, it's programmed cell death. So cell dies, okay, within its program, that 120 days, after 120 days it will go and die. Where it will go and die? It will go and die in the liver and spleen. So RBCs are autolyzing themselves in the liver and spleen, and liver and spleen is termed as the graveyard of RBCs. Liver and spleen is termed as the graveyard of RBCs. Okay, is the graveyard of RBCs. Who? Liver and spleen. Autolysis, that means they're going to die after 120 days. So what do the RBCs basically consist of? The RBCs basically consist of hemoglobin. Okay, now have a look at this word. It's a technical word, right? Heme is the iron part and globin is the protein part. Okay, globin is the protein part. Okay, so even when the RBCs die, this hemoglobin will split into heme and globin, out of which globin is a protein, so it will be reutilized, but heme will be thrown out of the body. But it will not be directly thrown out of the body. Heme or iron will be converted into bilirubin. Who's going to do this? The liver and the spleen. They are having certain mechanism along with the bile, okay, the gallbladder. We are going to convert iron to bilirubin to biliverdin. Okay, and this bilirubin, biliverdin together, they are giving the color to the fecal matter because it's excreted in the fecal matter. Okay, so the yellow color of the fecal matter is because of bilirubin and biliverdin. Okay, moreover, this bilirubin, biliverdin is conjugated. Conjugated, that means it is combined with proteins and then taken through the to the large intestine for excretion into the fecal matter. It's not let open anywhere in the blood, only except for in a disease, like in a disease called as jaundice. In that jaundice, what happens? This conjugation of bilirubin and biliverdin is open. It gets opened, so they are no more conjugated. Who? The bilirubin and biliverdin are not more anymore conjugated. They are let free, and when they are let free, they are moving all around in your blood and they are causing yellowness. What happens in jaundice, right? Your eyes become pale, your tongue becomes pale yellow, your skin becomes pale yellow, because bilirubin and biliverdin are going to all parts of your body, right? They're going to all parts of your body. So that is what is the hemoglobin and the digestion of hemoglobin. But what role does the hemoglobin have? The short form for hemoglobin is HB. Okay, so what role does the hemoglobin have? It is having the role to transport or carry oxygen. What role does it have? It is having the role to transport or carry oxygen. Okay, so whenever you inhale, right, the oxygen goes into your lungs. To be specific, it goes through your nasopharynx. From the nasopharynx, na, right, this is your, I'm not very good at drawing, but this is your nasopharynx region, right? Nasopharynx, then you enter your larynx. Okay, here is your larynx, here is your trachea with the C-shaped rings. You can feel over here, you can feel over here in your own throat, right? They are the trachea. That is the trachea, that is the C-shaped rings. Okay, those C-shaped rings will carry air further into the lungs, right? Now here the it is just behind, this is called as the larynx area, and in your larynx area we also have the sound box, right? You can just keep your hand over here and speak something. Say hello, right? Say hello. Keep, just do it right now, right? Keep your hand over here and say hello, right? You'll be able to feel some vibration, right? So that vibration is there of the larynx of the sound box. The sound box is something like this, an empty box which is having air and vocal cords. These vocal cords are longer in males and shorter in females. So hence the males are having a hoarseness in voice, whereas the females are having a high pitch voice. You've seen opera, right? So opera is that there is a glass, there is a lady who's kind of screeching kind of sound she's producing, right? And this glass will match with the frequency of her sound, and the glass breaks because of something called as resonance of sound, right? The amplitude of her voice and the frequency of her voice and the frequency of the glass matches, and then there is vigorous vibrations, and because of which the glass breaks. That you're going to have that in physics. Okay, so that is called as resonance of sound. Resonance is different in chemistry. Again, chemistry resonance is different, physics resonance is different. Physics resonance is what I've just told you, that is sound. Even when soldiers are walking on the bridge, they are asked to break the steps, because if the frequency of their marching matches with the frequency of the bridge will vibrate vigorously and it may collapse. So that is the resonance of sound. Well, anyway, this we are talking about the nasopharynx region. You inhale air, nasopharynx, pharynx, larynx. Larynx is having the sound box. This will further divide into two. All right, this will further divide into two. This is called as bronchi here, right? This will further divide into two. Yeah, two bronchi, and they will further divide and form alveoli. So alveoli is something like this, right? Alveoli, I'm bad at diagrams, but you'll understand. So this is something somewhat the alveoli, right? And alveoli, there are multiple alveoli inside the bronchi. Okay, and when this alveoli, when you're inhaling oxygen, the oxygen comes into the alveolus and gets diffused from here into these blood capillaries. Now once the oxygen comes into the blood capillaries, this oxygen combines with the hemoglobin to form oxyhemoglobin, and whatever carbon dioxide is coming into the blood, I mean the carbon dioxide which is released by the cells is coming to the blood, that will go into the you can say alveoli. But who carried that carbon dioxide? So 15% of carbon dioxide was carried by hemoglobin, and that was called as carbaminohemoglobin. Your 85%, okay, 85% of carbon dioxide is carried by bicarbonate. Okay, the bicarbonate ion carries 90% or 85% of your carbon dioxide. This bicarbonate combines with the H plus ions. Like whenever you eat any acidic things, sir, we don't eat acidic things. Of course you eat acidic things. Your tongue loves acidic things. You know why? Because see, whenever you are eating simple dal rice, or you can say as a boring sabzi and a roti, so what you are having, what do you ask mama? Mama, can you give me a pickle, achar? Why do you eat pickle? Because pickle is sour. The right pronunciation for sour is sour, and when it is sour, it is what? You are having H plus ions in that, right? It's acidic. All acidic things are sour to taste. Okay, and that sour thing, when you eat, you are indirectly eating H plus ions. Or when you are standing on the road and having pani puris, right? Those pani puris are also sour to taste, and obviously they are acidic. So does this acid alter the pH of our blood? Does it change the pH of our blood? It does not change the pH of our blood, because we are having a buffer in our blood, right? Even if you eat 20 pani puris, it's not going to change the pH of your blood. The pH of your blood remains constant between a range of 7.24 to 7.4. This is slightly alkaline. This is the pH of blood, which is slightly alkaline. So this H plus ions, whatever pani puri or whatever achar you are eating, will give you H plus ions that will enter your stomach. They may enter your blood, and there is buffer waiting over there just to combine with the with the H plus ions. See, H plus ions, if they are not let free, they will not be able to cause the acidity. H plus ions will cause acidity only if they are let free. So these H plus ions quickly combine with bicarbonate to form H2CO3, that is carbonic acid, and this carbonic acid comes to the lungs and it splits. Okay, it splits into H2O and CO2, and that's why with your every breath, every exhalation, you are exhaling water vapor and carbon dioxide, right? Some people who are wearing glasses can do that kind of thing, and they release some water vapor on the screen, on the glass, and they are able to clean that glass. And you know very well that we are releasing carbon dioxide out of our lungs. That means we are exhaling carbon dioxide in every breath. So oxygen, when it combines with hemoglobin, it's called as oxyhemoglobin. Carbon dioxide, when combines with hemoglobin, is called as carbaminohemoglobin. Although this is just 15%, hemoglobin is just carrying 15% of the carbon dioxide. 85% of the carbon dioxide is being carried by the bicarbonate or the carbonic acid. All right. So carbon monoxide, when it combines with hemoglobin, this is deadly. Okay, if you inhale carbon monoxide, and if that carbon monoxide combines with the hemoglobin, it is having 300 times more affinity with hemoglobin, and it is called as carboxyhemoglobin. Okay, carboxyhemoglobin. And this carboxyhemoglobin is like 300 times more stable than oxyhemoglobin, and because it is 300 times more stable than oxyhemoglobin, it does not leave the hemoglobin. Once it catches hold of the hemoglobin, it does not leave that hemoglobin, and all hemoglobin, once they get saturated, you are going to die. Why? You're going to die of suffocation. That's called as asphyxia, right? Asphyxia is death due to suffocation. Okay, and before that, your cells will die due to lack of oxygen. That is called as ischemia. Ischemia, whatever you want to call it as. And asphyxia is the death of a person due to lack of oxygen. Ischemia is death of cells due to lack of oxygen. Okay, so first thing, ischemia is going to happen because your cells will stop getting oxygen, because all the hemoglobin who is combined with all the hemoglobin, carbon monoxide is combined with all the hemoglobin. That's why it's said that don't park your car in a garage and sit in the garage with the AC on, because that AC, if it is pulling the air from outside, it will pull in the carbon monoxide, and it will cool that carbon monoxide and send it inside the cabin of the car. And this has actually happened. It's an accident which has happened, which caused the death of the driver, because he was sleeping in the car inside the garage with his AC on, and the car was also running. So it pulled in carbon monoxide. When the car is standing at one position, it releases carbon monoxide instead of carbon dioxide. Whenever, like, because it's when it is standing, when it is idling, it's called as the idling position. So when it is idling, it is not completely burning the fuel, and that's why it releases carbon monoxide in place of carbon dioxide. And because of which, carbon monoxide combines with your hemoglobin, forms carboxyhemoglobin, does not leave your hemoglobin, and eventually causes death due to ischemia and asphyxia. Okay, then cyanide if combines with hemoglobin, it is called as methemoglobin. Right, what it is called as methemoglobin. So methemoglobin is cyanide combining with hemoglobin. Okay, so these are certain terms. So let's come back to what we were doing. RBCs, the function of RBCs is to transport or carry oxygen, right? So one more thing, that the RBCs are biconcave in shape. What are they? They are biconcave in shape. Biconcave meaning they're having a depression on the top as well as the bottom, so that these RBCs can enter the finest of capillaries. The blood capillaries are very fine, very thin in diameter, and so the RBCs should be able to enter the finest of capillaries and try to give oxygen to each and every cell of the body. That's why they are made biconcave. And for them to become biconcave, they are devoid of two things. Devoid meaning they are two things are absent in them. They are devoid of, number one, mitochondria and nucleus. So if there is mitochondria in the RBC, the RBCs will themselves start consuming the oxygen which they have to give it to others, to give it to other cells. जैसे कि आप चोर के हाथ में तो पैसे नहीं देते ना, कि भाई ये पैसे आप जाके बैंक में जमा करो, तो वो बैंक में नहीं जमा करेगा, खुद के पॉकेट में जमा करेगा. सो चोर के हाथ में जैसे पैसे नहीं दिया जाते, वैसे ही ऑक्सीजन अगर माइटोकॉन्ड्रिया इस आरबीसी के अंदर हो, तो आरबीसी वो ऑक्सीजन दूसरे सेल्स को देने की जगह पे इट वुड गिव, it will consume that oxygen itself. And that's the reason why we are avoiding, I mean, the RBCs are devoid of mitochondria. Okay, and devoid of nucleus also. Why it's devoid of nucleus? Because see, one thing, like RBCs don't require energy. They are propelling with the flow of blood. They're getting propelled with the flow of blood. They don't have to move on their own. They don't have to spend energy. So even if they don't have mitochondria, it's okay, and they will not consume oxygen. Number one. Number two, if they don't have nucleus, see, nucleus they don't require because they're not going to divide. Nucleus is required for cell division. Basically, it has the genetic material. So they're not going to divide, because new RBCs are formed from a stem cell which is called as megakaryocyte. Megakaryocyte is the stem cell which is producing all the blood cells. See, stem cells are totipotent. They can form all different kinds of cells, right? In plants, all cells are totipotent, but in humans, only the stem cells are totipotent, and only they are able to, only megakaryocyte is able to produce all kind of the WBCs, RBCs, all kind of blood cells. Okay, so RBCs need not have the nucleus because they're being produced by the megakaryocyte. So because they are devoid of mitochondria and nucleus, it is helping the RBCs to become what? To become biconcave in shape, to reduce its size so that it can enter into the finest of blood capillaries. Now, finally, the RBCs are the number of RBCs. How many number of RBCs are there? So there are around 4 lakh, or you can say 3.5 lakh. Okay, 3.5 lakh to 4.5 lakh RBCs per ml of your blood, or per centimeter cube. That means per ml of your blood. There are so many RBCs per centimeter cube, that is per ml of your blood. Because RBCs are very small, you can fit in more RBCs in 1 ml of your blood, and they are required. Those many are required because they are carrying oxygen to each and every cells of your body. You know that you are having billions of cells in your body, right? And then coming to the leukocytes. Leukocytes, they are not having such a huge number. They are only 5,000 to 10,000 per ml, because they are large in size. They're pretty large in size as compared to the RBCs. Okay, so these are 5,000 to 10,000 per ml. What? The WBCs. I'll not be able to fit in WBCs over here, so I'll write WBCs separately, and let me classify them separately. Okay, so all this we have seen. Let's go to WBCs. So WBCs, also called as the leukocytes, can be further classified as granulocytes and agranulocytes. We can classify them as granulocytes and agranulocytes. And the lifespan of RBCs was 120 days, but the lifespan of WBCs is very less, 8 to 10 days. And the number of WBCs I've told you, they are 5,000 to 10,000 per ml of your blood. So granulocytes. Now granulocytes can be further divided into, yeah, acidophils, which is also called as eosinophils. Now you need to understand this, that acidophils, why they are called as eosinophils? Because they are being stained. Okay, they are being stained by a dye. Which dye? Eosin is the name of the dye. It's an acidic dye. Acidophils is the cells which love acidic pH. Okay, the cells which love acidic pH is called as acidophils. Okay, and which is that acidic dye which they love? That is eosin is the dye which they love. They get stained by eosin. Okay, then we are having basophils. Basophils are, they obviously get stained by a basic dye, and they love the basic pH, that is methylene blue. And then we are having lastly the neutrophils. Okay, the neutrophils, they are obviously they like the neutral pH, and they are the largest in percentage. They are 62% of all the WBCs. Okay, so they are having a larger percentage, and the function of the neutrophils is to perform phagocytosis. The function of the neutrophils is to perform phagocytosis. I hope you know what is phagocytosis. If there is a bacteria over here and there is a WBC over here, this WBC engulfs this bacteria. It changes its shape such that the bacteria comes over here, and it literally eats up the bacteria. It eats the bacteria and destroys it. This process is called as phagocytosis, eating of the bacteria. Okay, then cell drinking is called as pinocytosis. Cell eating, phagocytosis. Cell drinking, pinocytosis. Okay, so the neutrophils basically perform phagocytosis. Then basophils are also called as the mast cells. Okay, they are also referred to as the mast cells, which store histamine. Okay, now when is this histamine released? So histamine released in the stomach is different, released in your skin is different. Okay, so histamine when released in your on your skin, when is it released in your skin? When you are having a bee sting. If a honeybee stings you, that area becomes red. It becomes a little swollen, inflamed, and it shows radiation of heat, right? So wherever histamine is released, see, even during common cold, right? Common cold, histamine is released in your nose. How does the nose become? The nose becomes red, right? And what happens, there is slightly inflammation, halka sa swelling aa jata hai naak par, so it's redness is there, inflammation is there. And see, even though you're sneezing or whatever is happening, your nose is running nose, meaning it is what it is. Running nose does not mean that your nose is running somewhere. Running nose meaning it's continuously secretions are coming out of your nose. Okay, that's called as rhinitis. In biological terms, that's called as rhinitis. So if histamine is released in your nose, it's going to cause rhinitis. It's going to cause the redness in your nose, and आपको गरम गरम लगेगा. आप ध्यान देना, it गरम गरम लगेगा आपको. That is what radiation of heat. Radiation of heat. And there is also inflammation meaning the swelling. The other word for inflammation is swelling. So नाक में histamine release हो गया है, तो there is swelling, plus there is radiation of heat, गरम गरम लग रहा है, and there is redness. Same thing happens when there is a bee sting. एंड सेम थिंग हैपेंस अगर जोर से कोई कान के नीचे बजा देता है, ठीक है, कान के नीचे बजाने के बाद में थोड़ा देखना, ध्यान देना, ठंडा लगता है, गर्म लगता है, ऑब्वियसली गर्म गर्म लगता है. दैट इज रेडिएशन ऑफ हीट, बिकॉज देयर इज हिस्टामाइन बीइंग रिलीज्ड ओवर देयर. द मास्ट सेल्स आर रिलीजिंग हिस्टामाइन. There is radiation of heat, there is inflammation, there is redness. Okay, कभी कभी हाथ के निशान भी आ जाते हैं, तो उसको क्या कहते हैं? वर्ल्स. जब आप मार्क्स अच्छे नहीं लाते हो, जो पेरेंट्स जो आपको प्रसाद देते हैं, दैट इज वर्ल्स. वर्ल्स मतलब हाथ का निशान यहाँ पर आ जाना, राइट? सो दैट इज व्हाट? हिस्टामाइन. Okay, histamine or mast cells, right? Mast cells released by the or histamine released by the mast cells, right, which are also called as basophils. So their function is allergy. They work in allergic conditions. Histamine and all that is working in allergic conditions. Sometimes histamine is also released because of inhalation of pollen grains, or maybe some flower particles, some particles, dust you have inhaled. Some people have that allergy. It is because of their extra, you can say, sensitive basophils. Then acidophils or eosinophils, what are they responsible for? Yes, what are they responsible for? No, that is neutrophils, no? Phagocytosis. They're responsible for what? Yeah, but the function? No, acidophils. Yeah, so engulfs bacteria is nothing but this thing only, phagocytosis only. Yeah. Is there any other function? Secretes what? Antitoxin. Yeah, secretes antitoxin is another function of acidophil. Secretes antitoxin. Okay, so this is your entire classification of granulocytes. We have finished the now why they are called as granulocytes, because their cells are having granules. They are granular in nature, and their nucleus is also typical. They are lobed, bilobed. You need to check out that, right? They are granular. Acidophils, basophils, neutrophils, all the three are granular. Who are non-granular or agranulocytes are the two cells, that is monocyte and lymphocyte. Monocyte is the largest WBC, and lymphocyte is the WBC which is important in increasing your immunity by producing antibodies. What does it do? Produces antibodies, right? Produces antibodies. That's why it's very important, has a very important role in immunity. And the monocytes are the largest WBC. As I told you, they are the largest WBC. Who? Monocytes. And the function of monocytes is again phagocytosis. Function of monocytes is again phagocytosis. For remembering the lymphocytes, right, that which antibodies do they produce? So which antibodies? Then again, if you want, there is, these lymphocytes can be further divided as B and T lymphocytes. If you want to go in detail, this is produced in bone marrow, B lymphocyte, right, produced in the bone marrow, matures in the bone marrow. And T lymphocyte is produced in the bone marrow but matures in the thymus. Where does it go and mature? T stands for thymus. Thymus is another important organ of the lymphatic system. And B lymphocytes is they're maturing in the bone marrow. and they are maturing in the thymus. That's why B for B lymphocytes, bone marrow, T for thymus, that is the T lymphocytes. Okay, now which are the different types of antibodies? Now just remember a mnemonic for remembering the antibodies. Okay, gamed is the mnemonic. How do you remember the different antibodies? It is gamed. It is IgG, immunoglobulin gamma, immunoglobulin alpha, IgA, IgM, immunoglobulin mu, then IgE, immunoglobulin E, and IgD, immunoglobulin delta, immunoglobulin epsilon, immunoglobulin mu, immunoglobulin delta, immunoglobulin A alpha, immunoglobulin G gamma. Right? So out of these, IgM is the largest antibody. IgM is the largest lymphocyte, largest antibody. Okay, and IgA is the smallest antibody, which is transferred from mother's milk to the fetus, and not any milk. It's the first milk. Okay, first milk, which is called as colostrum. The first milk which is released by the mother for the fetus is rich in antibodies. It is having which antibodies? Is having IgA antibodies, and the first milk of the mother, which is rich in antibodies, is called as colostrum. So first milk has to be given to the fetus because it is rich in antibodies. That's the first acquired immunity which the child is receiving from the mother, right? So IgA, it's the smallest antibodies. IgM, the largest of the antibodies. IgE released during allergy. IgE is released during allergy. Okay, so these are gamed. Just remember, gamed is the mnemonic for remembering the different antibodies. So here we have finished the WBCs in detail, right? And we have also finished about RBCs also in detail, right? And now the last part which is remaining is thrombocytes or platelets. So thrombocytes or platelets, they are around 150,000. Okay, 150,000 to 250,000, meaning you can also call it as a 1.5 lakh, 1.5 lakh to 2.5 lakh per ml of the blood, per centimeter cube or per ml of the blood. Okay, now they are involved in basically they are involved in the mm, blood clotting, and blood clotting is a separate topic. Now it's a big topic, blood clotting, so I would prefer recording it in the next session. Okay, that is blood clotting. It is having a series of factors. It's very interesting to note how it is forming fibrinogen, fibrin, and finally it forms a mesh and clots the blood. All that is very interesting. Wait for that in the next class. And there is a series of factors, so we'll be doing in ultra detail. We'll have one separate video on clotting of blood. Meanwhile, I hope you all have understood what is the classification of blood, how different cells are playing different roles in our body, what is plasma, what is sera or serum. All this we have covered in today's session. So do not forget to share, like, and subscribe, and let me know in the comment section how you liked or enjoyed the video, or if you are having any suggestions, please do let me know. Thank you.